Bismillahirrahmanirrahim. We are going to discuss the MCQs in microbiology. Today we will focus on the general microbiology part 1. In the coming lecture, we will discuss the more about the general microbiology MCQs. The very first MCQs is why is fungus called a eukaryotic microorganism? Is there are two types of the organism or cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells only include the bacteria or kingdom Monera. Fungus is a eukaryotic. What characteristics that include the fungus in the eukaryotic organisms? What is the best answer amongst these? So the option A, that is the fungus has cells that have the nucleus. The most important difference between the prokaryotic and eukaryotic is the presence of the membrane-bound nucleus. Nucleus envelop cytoplasm and the organelles. So the answer is A. What is Archaea? Archaea is a prokaryotic microorganisms. It is the third domain of the living organisms, but Archaea is somewhat differ from the prokaryotes bacteria. What characteristics that differentiate the Archaea from the bacteria? What is the best answer amongst these? So Archaea is the prokaryotic organism that have cell wall, but its cell wall has no peptidoglycan. So option number C is the best answer. Archaea is a classification of organism that identify prokaryotes. It is a prokaryotes, but they do not have the peptidoglycan cell wall. Cell wall is present, but cell wall is not made up of peptidoglycan. Its cell wall is made up of a pseudopeptidoglycan. So option number C. What is phagocytosis? It's a process that is used to engulf the foreign particles by the phagocytic cells, white blood cells, macrophages, monocytes. So the answer is C. The ability of a cell to engulf and digest solid materials by use of pseudopods or false feed. This is the definition of the process of the phagocytosis that engulf and digest the solid material. What's a compound microscope? There are two microscopes, two types of microscope. One is called a simple and the second is called a compound microscope. So simple microscope can be differentiated by the compound microscope on the basis of the sets of lenses. Simple microscope has a single lens while the compound microscope has a two lens. So option number B is correct. A microscope that has two sets of lenses, an ocular lens and an eyepiece, that is a compound microscope. What is germ theory of disease? Germ theory of disease mainly focus on the ability or the role of the microorganism in the infection. Germ theory mainly explains that there is a specific microorganism cause a specific infection. That particular type of infectious agent or pathogen must be found in the infected or diseased person. So we select the best answer amongst these. This is the option number A. Germ theory states that this is a definition of the germ theory. Germ theory states that a disease causing microorganism should be present in animals infected by the disease and not in the healthy animals. The organism that is responsible for the infection for the disease that must be present in the infected organism, and it is not present, it is not found in the healthy organism. There is a strong link between the infection and the microorganism. What is Edward Jenner's contribution to microbiology? Edward Jenner is a scientist in the 18th century. The most important contribution of Edward Jenner in the microbiology is towards the role in the process of the vaccination. So the best answer is B. Edward Jenner discovered how to create vaccination to trigger the body immune system to develop antibodies that fight microorganisms. He used the cowpox organism to protect the individual by the smallpox. The fact that viruses are obligate intercellular parasites means that they require a dash for reproduction. What require is viruses are obligate intercellular parasite that must require a host for their reproduction. The host is the best answer. Which event came first? From the history. Koch demonstrated that the bacterium anthrax can cause disease, or physio measure the velocity of light. So the physio measure the velocity of light before as the Koch postulates. Which event came first? Watson Crick described the DNA double helix, or first hydrogen bomb exploded. So first hydrogen bomb exploded first before the Watson Crick described DNA double helix. Which event came first? Neil Armstrong walks on moon before the discovery of the Lyme disease. Who first described microorganisms such as bacteria? The first scientist who described the microorganism that is mainly known, the father of microbiology, that is Antony von Leeuwenhoek. Who first developed the process of colony purification on solid media? Robert Koch was the first scientist who uh, developed the process of colony purification of the solid media. So it was the Robert Koch. Who was the first successful solid what was the first successful solid medium for colony purification of bacteria? It was a potato. Who first suggested using the thickening agent most commonly used for colony purification today? It was Fanny Hess. From the history, Fanny Hess. The most important innovation, new idea in Pasteur's swan's neck floss experiment was the best answer. The fresh air could directly contact the medium. Fresh air could directly contact the medium. Credit for the first vaccine for prevention of human disease is generally given to Edward Jenner for the prevention of smallpox. To make a vaccine against the chicken cholera that would not kill the chicken pasture, attenuated the strain by repeatedly passaging it in culture. उसने he repeatedly culture the pathogen in the attenuated repeatedly culture, and it was not able to cause the disease, but it can boost the immune system. When Louis Pasteur first tried his vaccine on young boy, there was possibility that the vaccine itself could kill the child. This was permissibly under the standards of day because what was the reason? The child had rabies, which was always fatal. उस child को rabies थी जो भी एक जो fatal disease है fatal infection है. Agostino Bessi first showed that a disease like silkworms was caused by microorganism. What kind of microorganism caused the disease? It was caused by the fungus, which was caused by the fungus. What was the first bacterium shown to cause human disease? The first bacterium जो identify की गई उसका नाम है anthrax by the Robert Koch. The primary use of Koch postulate is to give the different postulates. The primary use of the Koch postulate is to demonstrate that a disease is caused by a microorganism. Disease is caused by the microorganism, a role of the microorganism in infection or the disease. What was the first virus shown to cause the disease? The first virus that was identified, the first virus their role was played in the infection was the tobacco mosaic virus. Which of the following is not a part of Koch postulates? Koch have given different types of postulates. Amongst these we select the postulate that is not the part of the Koch postulates. The microorganism must secrete toxin in culture. It is not necessary to cause to produce toxin in the culture. The role of blood cells in fighting disease was first demonstrated by Meshnikov with his observation of phagocytosis. Meshnikov with his observation of phagocytosis. The role of antibodies in fighting disease was first demonstrated by injection of rabbit antitoxin to protect against the diphtheria. It was the first role of antibodies in fighting the disease. Rabbit antitoxin to protect against diphtheria. The first observation that bacteria-like organisms could be found in normal air was by Louis Pasteur. Louis Pasteur gave the first observation that bacteria could found in the normal air. The first physician to make practical application of the germ theory of disease to surgery was Joseph Lister. Joseph Lister was the first physician to make the practical application of germ theory of disease to surgery. He gave the antiseptic process. Which of the following discoveries is not attributed to Sergei Vinogredsky? A colony isolation on solid phase medium was a not contribution. If colony isolation on solid phase medium is not the contribution of the Sergei Vinogredsky. Use Pasteur's studies on the unwanted production of acid from beet sugar was the first demonstration that bacterium can cause specific chemical reactions. Bacterium can cause specific chemical reactions. Spirulina belongs to which group? It is the Xenophyceae, Cyanophyceae, Rhodophyceae, the Phaeophyceae. So the best answer is the Cyanophyceae. The first antibodies to contact invading microorganism was IgM was the first antibody that produced against when a body contact with a pathogen. So IgM is the first antibody produced against the infection. The light emitted by luminescent bacteria is mediated by enzyme that is the luciferase enzyme is produced by the luminescent bacteria. It is produced by the firefly. Food poisoning is caused by which organism is responsible for the food poisoning? Clostridium botulinum is responsible for food poisoning. Copes, uh, Cope-like spots will develop in the measles. Viral DNA is resistant to DNA of the host cell bacteria. It contains 5' HMC group. Which of the following is an example of live vaccine amongst these? Which is the live vaccine? Cholera is a live vaccine. All other are the killed vaccine. Triplet toxoid vaccine gives protection against. It is used for the three different types of the pathogens. So these are the for whooping cough, that is called the arboreal pertussis, tetanus, custom tetani, and the coinhibitum diphtheria, that is called the diphtheria. So uh, DPT is the vaccine used for three different types of infectious agents or three different infections. Higher doses of chlorphenicol affects the eukaryotic cells because they have mitochondria. Is chlorophenicol affects the ribosome and mitochondria has its own ribosome just like the ribosome of the bacteria. AIDS is caused by the retroviruses, HIV viruses. Penicillin is a primary metabolite or secondary metabolite, tertiary, so it is a secondary metabolite. The organism in alcohol fermentation is Aspergillus niger, Bacillus subtilis, Saccharomyces cerevisiae, or Escherichia. So it is the answer C, Saccharomyces cerevisiae. Alcohol fermentation में mainly Saccharomyces cerevisiae इस्तेमाल होती है. Which vitamin B12 can be estimated and determined by using algorithm? It is the Lactobacillus leishmani. Lactobacillus leishmani can use to determine to estimate vitamin B12. Batch fermentation is also called closed system fermentation. To differentiate lactose and non-lactose fermenters, the medium used is the Mechaniki medium is used for the differentiation of lactose and non-lactose fermenters. The microorganisms used full for fermentation are which are mainly useful for fermentation. The yeast is mainly used for the fermentation. Industrial microbiology mainly depends on the phenomena of process of the fermentation. Streptokinase is also termed as a fibrinolysine. Streptokinase is also known as a fibrinolysine. Streptokinase is produced by Streptococcus pyogenes. Large vessels containing all the parts and conditions necessary for the growth and desired microorganism is called the bioreactor. It is used in the process of fermentation. Basic principle in industrial microbiology is suitable growth condition, fermentation, providing acidic condition, all are good, but the fermentation is the best answer. is the basic principle. The discovery that led to the development of first antibiotic was made by the scientist Fleming. Spirulina is it is an edible fungus, biofertilizer, biopesticide, a single cell, which is a single cell protein. Biogas is both A and C are correct. Methane rich and propane rich fuel. Higher value of biological oxygen demand (BOD) shows that it is what is highly polluted. What is highly polluted? Which of the following is fermentation process? Both A and B are fermentation process. Batch process and continuous both are the fermentation process. Who showed that Saccharomyces cerevisiae causes fermentation forming products such as the beer and uh, buttermilk. It was the Louis Pasteur. Rennet is used in the process of the fermentation, bread making, cheese making. So mainly it is used for the cheese making. Tissue plasminogen activator. It is a vitamin, an enzyme, or chemical that's not sugar differentiation or amino acid. So a tissue plasminogen activator is an enzyme. A bioreactor is a reactor that is used as a fermentation tank. Fermentation tank. Hemoglobin is a protein. Which of the following can be application of fermentation? So amongst these, which is the application? So all of these are the application. Tanning of leather, curing of tea, and all are the application of fermentation. Enzyme immobilization is providing enzyme with a protective covering. Option B is the correct, providing enzyme with protective covering. Biogas is produced by both B and C are correct. Anaerobic breakdown of biomass and with the help of methanogenic bacteria. Name the first organic acid produced by microbial fermentation. It was a lactic acid. It was a lactic acid by Lactobacillus bulgaricus. Vinegar is obtained from molasses with the help of both B and C. Acetobacter and yeast are used for the vinegar formation. Acyclovir is used for the treatment of the herpes simplex viruses. It is an analog for what? So it is a guanylate derivative. So it is can used for the guanylate GTP, guanosine triphosphate analog. So it is a guanine derivative. Sterilization is a process that is used for the removal of all organisms, including the spores. The definition of the sterilization. MRSA, methicillin-resistant Staphylococcus aureus. MRSA is mainly include the microorganisms that do pulse field electrophoresis, is useful in the investigation of the outbreak. Option number D, and is sensitive to vancomycin. Both option D and E are correct for MRSA. Action of following antibiotics are correct. Sulfonamide interfere with the folic synthesis. It's a correct. A option is correct. Metronidazole interfere with DNA synthesis. Option B is correct. And chlorphenicol inhibit protein synthesis. A, B, and C both are the correct option. Endotoxins are the toxins that elicit an antibody response which may protect the host from the future attack. Option number B. And can often survive in autoclaving. Option number C. Both are correct. Stains useful for identifying fungus include both C, D, and E, hematoxylin and the eosine, and the gymori, methanamine silver, and the periodic acid shift that is called the past. Both C, D, and E option are correct for the identification of the fungus. The identification of bacteria by serological tests is based on the presence of specific antigens. Which of the following bacterial components is least likely to contain useful antigens? So amongst these, ribosome is least likely to contain useful antigens. All the others have the antigen. Cell wall has antigen. Capsule also acts as antigen. Flagella also acts as, but ribosome has the least likely to use as a antigen. Each of the following statements concerning the gram stain is correct except which option is correct? Escherichia give the pink color, it's the correct. Streptococcus pyogenes give the blue color, it's a gram positive correct. Now, mycoplasma pneumonia is not visible in gram stand because it has no cell wall, and the best answer is the Mycobacterium tuberculosis stands blue because it has a thick lipid bilayer. Microbacterium mainly not identified by using the Z by using the gram stand. It mainly is identified by using the Z and staining. That is also called the acid fast staining. So, an outbreak of sepsis caused by the Staphylococcus aureus has occurred in the newborn nursery. You are called upon to investigate according to your knowledge of the normal flora. What is the most likely source of the organism? It was from the nose. Each of the following organisms is an important cause of urinary infection except it was the Bacterial fragilis is not responsible for the uterine infection mainly. A 30-year-old woman has non-bloody diarrhea for the past 14 hours. Which one of the following organisms is least likely to cause this illness? So, the organism mainly Streptococcus pyogen is not responsible for the pyogens, not responsible for the diarrhea. Which of the following agent is a recognized cause of diarrhea except? The best answer is Enterococcus faecalis is not responsible for the diarrhea. Each of the following statements about the classification of streptococcus is correct except wheat and streptococci are identified by Lancefield grouping which is based on the C carbonate in the cell wall. It is not the correct answer. Thank you so much. We have discussed the general MCQs in the microbiology. In the next lecture, we will discuss the more general MCQs in the microbiology. Then we will focus on the further bacteriology, immunology, and medical biology as well.